Welcome to Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're looking at being able to recognize the organelles from electron microscope images. So electron microscopes produce high detail images of animals and plant cells, and this is the result of higher resolution than the light microscope. Resolution can be defined as the ability to distinguish between two separate points. The resolution of a light microscope is lower than an electron microscope, as it is limited by the wavelength of light. Electron microscopes rely on electrons, which have a much smaller wavelength than visible light. Electron microscopes can be used to study specimens bigger than 0.5 nanometers, and the microscope fires a beam of electrons at the specimen. The electrons are picked up by the electromagnetic lens, which is what generates an image. However, the specimen used must be dead, and there are two types of electron microscopes you should know about. So there's the transmission electron microscopes and the scanning electron microscopes. So you might have gone through these briefly in GCSE, but you need to know more in A-level. So transmission electron microscopes use electromagnets which focus on a beam of electrons. And in this, so for this microscope, the specimen has to be thin. This beam is transmitted through the specimen, and the denser parts absorb more electrons, hence the denser parts appear darker in the image. And the kind of images you get from a transmission electron microscope would be 2D images. So now we're looking at scanning electron microscopes. So a beam of electrons is scanned across the specimen. The specimen does not have to be thin. The beam bounces off the surface of the specimen and the electrons are detected to form an image. And in this case, so for scanning electron microscopes, a 3D image is formed. However, the resolution is much lower than transmission electron microscopes. So how do you recognize organelles from images generated in a microscope or just your diagrams in general? So one way to revise for this would be to use the diagrams you see in your official textbook because, well, that's the one they're most likely going to ask and it's got all the labels on it. However, you do have some obvious clues that might suggest what different structures are. So if you see a spherical structure, it's usually the nucleus, and the darker circle in the nucleus would be your nucleolus. So you need to know the distinction. If you see the Wi-Fi symbol-like structure, that's usually the Golgi apparatus because the structure is more uniform. There's like a set pattern. If you see spots on some specific structure, then that's most likely your raphia. And if it's smooth and not so uniform like you would notice in the Golgi apparatus, it's going to be your smoothia. And cylinder-like, I wouldn't say cylinder, but like an oval-shaped structure, that's usually the mitochondria. And one more thing is that if you're looking at a plant cell diagram, the outer layer is going to be your cell wall. It's not your cell membrane, it is the cell wall. However, if it's the diagram of a bacteria, then it would be the capsule. So those are like different things to look out for. And finally, another very important way to revise this topic is through past paper questions because the official textbook is going to have like maybe the pictures are in color or maybe the pictures are very clear, but then the past paper questions, the images aren't going to be that clear, and that's what you're going to see in the actual exam. So to get used to that, you need to do a lot of past paper questions. So that is all from Professor SNAB today. Hope you found this video useful. If you have any questions, please let me know in the comments below and I will get back to them as soon as possible. And don't forget to like, share and subscribe.